Good morning, everyone. As Ellie said, I'm a PhD student in the Innate Immunity and Inflammation Lab at IGC, and I'm here to present you some preliminary data that we have on a project that we've been exploring in the lab. So our lab is one of the main focus of the lab is sepsis. We can easily define sepsis as a dysregulated host immune response to infection. This has a high incidence worldwide, and it has a high elevated death rate associated. Most of the treatment options out there are focused on clearing the pathogen, and in an alarming percentage of the cases that doesn't improve the outcome. So new therapeutic approaches are required. In these new therapeutic approaches, there are one that is gaining traction, it's disease tolerance. So disease tolerance can be easily defined as approaches that reduce the damage associated with an infection without directly focusing on changing pathogen burden. There are several ways that we can do this. Our lab is focusing on how activating stress responses on a cellular level can lead to this disease tolerance. So the general theoretical framework is by causing a stress on an organelle within a cell, we lead to the activation of a response that is going to solve that stress and, bottom line, improve the cellular function. What we see is by causing this stress in the context of an infection, we can improve the cellular function, and that can, in fact, improve the outcome against sepsis through a disease tolerance framework. This approach has proven to be fruitful since now in the lab we've shown that by inducing DNA damage response as well as mitochondrial stress, we get an improvement of murine sepsis outcome in a pathogen burden independent way. As you can see from the titles of these papers, the way that we induce these stresses are via pharmacological treatments. And one of the questions that came up recently is what is the role of the gut microbiota in this? And why the gut microbiota? Besides being highly disruptive in sepsis, as Sorel just showed you, there are a number of non-antibiotic drugs that clearly influence the community. At the same time, there is also evidences of the gut microbiota heavily having a dependency or having an association with certain microbiota profiles. So we understand that it's important to understand the influence or the interplay of the gut microbiota on these disease tolerance-based therapeutical approaches in the context of sepsis. So the way that we got into this area of the gut microbiota was by doing some experiments with the Anthracycline story. We received the mice from the Charles River. We treat them under germ-free conditions to try to reduce as much as possible external influences on the gut microbiota. We would leave them to rest for 7 days, and after 7 days, we would do feces collection, we would do drug administration, and the infection with E. coli. Between day 7 and day 16, we would do survival. We would measure survival, temperature, and weight. So we took this setup and we did it 2 different times. And what we saw was interesting because on the first experiment, as you can see, we had a better outcome by treating the mice with epirubicin, one anthracycline. While in the experiment 2, we had a better outcome with the mice treated with actylrubicin. And so since we had the fecal content of these mice, we decided to do 16S amplicon sequencing. And what we saw was two different communities associated with two different better outcome with two different anthracyclines. We became a little bit paranoid, but we then realized that this difference was based on the fact that mice from two experiments were coming from two different rooms within the same facility of Charles River. But we decided to look into this further on to understand if there could be something in the profiles, on the gut microbiota profiles of these two Charles River room mice that could explain why we would have this difference. So after the beta diversity measures, we looked into the alpha diversity measures, and as you can appreciate here on the right side, we see that in terms of diversity through the Shannon index, the values are very similar between the two rooms, while for richness, both here present by the observed ASVs and the Chow index, we see a little bit a bigger richness for the mice that have this ACLA profile, which means that basically we can say that the ACLA profile mice have a richer community, which at a certain point makes sense because actylrubicin is one of the most toxic anthracyclines. So it's a sign of resilience, I would say, meaning you have a higher diverse community, and that helps you to react better to more toxic drugs. Then, regarding differential abundance, we saw specific ASVs across a set of families, but there were two specific families that caught our eye. Both of these families have the machinery necessary to detoxify one anthracycline that we didn't use in this assay. But they seem to have the machinery required to detoxify. And what they do is they detoxify, they create secondary metabolites that are as effective, but less toxic. So this could be an interest direct effect of the gut microbiota on the drug efficacy. At the same time, this first family, Huminococcaceae, has been associated with several drug efficacy across several conditions, immunotherapy, etc. So it's again one sign that this family is again important in another context. So from this super preliminary data, what we can see is there are specific gut microbiota profiles that are associated with a better outcome when treated with specific anthracyclines. What we are looking, if this is real, what we are looking is either a direct effect, meaning either the microbiota is metabolizing the drug or the drug is having a direct effect on the microbiota, or an indirect effect, which means the involvement of third parties. On a future perspective, we want to study the gut microbiota interactions with other drugs, specifically the ones that we used for the second paper that I talked about, the doxycycline, a tetracycline antibiotic that, again, has been shown to cause mitochondrial stress and does improve the sepsis outcome. In the course of that study, beyond the antibiotic effect, we saw that the doxycycline had a role on regulating one of the master regulators of the bile acid cycle called FXR. And by taking advantage of lock system. We removed the FXR from the liver and then infected them with E. coli. And as you can appreciate here, by removing the FXR from the liver specifically, there's an ablation of the effect of the doxycycline. So it would be interesting to see how by modulating FXR using doxy, for example, how that has an effect on the gut microbiota, considering that bile acids is one of the most important metabolites in the context of the microbiota. And also, another drug that is under our interest is fenformin, that it's an antibiotic drug from the D118 family, a family that has been shown that is both affected and affects the gut microbiota. So with this, I would like to thank all the funding agencies that made this project able to kickstart. This is still in a very early stage, as you can see. To thank IGC and specifically the IGC facilities to make all of this work possible, and to open the floor to questions and hopefully to collaborators to work on this project. Thank you.
Thank you, André. Are there any questions? Oh, I see one hand there. Katya. Is that a... Oh, no, it's clapping. Sorry. Yeah. Okay. There's a question from German. Please go ahead.
Thank you. Actually, it's two very quick questions. One is when you do the 16S analysis, are you, because there is always this Lactobacillus and Ruminococcaceae apart. Do you know how similar they are? Because I always wonder if it might be actually different copy numbers of the same genome. And the other one is also when you calculate the diversity, I would assume that the first effect that drugs have on the microbiota is to reduce the total bacterial loads. And that could complicate the comparisons of the diversity because the Shannon diversity is very similar. So I wonder if you have any indirect measure of total loads of the bacteria. Thank you.
So starting from the second one, this profile is before drug treatment. So there's no direct effect from the drug because the drug is still not in the system. These mice are completely unstimulated. So this is something that a priori they had that could influence the downstream, the specific treatment. Regarding the first question, that is a very interesting point that we haven't looked into. But since we are now focused in terms of this project to try and repeat and see if first we still see the same phenotype, because the gut microbiota in mice in specific facilities is varies a lot. And at the same time, if we see, and this is something that worries us, that by itself there's a gut microbiota profile that improves the survival without any drug treatment than the other. And so we are now trying to focus on that, but for sure, as soon as we nail down in these two families, if it's these two families, we'll sure have to look into that. Yeah. Thank you for your questions.